Welcome to the deep dive, where today we're going deep into the world of tweaking life at its most basic level,
Yeah,
specifically microorganisms and how scientists change their DNA to make them do all sorts of amazing things.
it really is rewriting those blueprints, isn't it?
It is. It is. And you sent me a bunch of really interesting articles on this,
It did.
and it seems like we can break this down into three main categories.
Yeah, I think that's a good way to think about it. We've got chemical methods, physical methods, and then the biological methods.
Right. So this deep dive is all about giving you the core
Data
ideas,
essentials, yeah.
some of the surprising little details about how these techniques work.
Those little interesting bits.
So first up, chemicals?
Chemical mutagenesis, that's where we start. It's basically using compounds that react with DNA
Right.
to change it in some way, or sometimes directly alter it.
Oh, so it's not always a reaction.
Sometimes it's just, a direct modification.
Huh. Interesting. So, what kind of chemicals are we talking about here?
Well, you've got your base analogs, for example.
Base analogs, those are the ones that kind of sneak in, right?
Yeah, they're kind of sneaky. They mimic the normal DNA bases.
Oh, so they trick the cell.
They do. It's like a wolf in sheep's clothing.
So what?
You've got things like, 5-bromoracil
Right.
and 2-aminopurine. They get incorporated during replication.
Oh, so they look like the normal building blocks of DNA.
They do, and that's how they trick the cell.
But then they cause problems later on.
Exactly. They cause mispairing during replication.
Wow, it's amazing how something so small
It really is.
can have such a big impact.
Yeah, it highlights just how precise these cellular processes are
Yeah.
and also how vulnerable they can be.
And then we've got those DNA modifying agents.
Right. Those directly go after the DNA bases.
So they're not pretending to be something else.
No, they're more like, a direct attack.
Much more aggressive.
Yeah, and there are alkylating agents in that category, things like EMS and NTG.
Yeah. Now, EMS, I remember reading that it tends to cause very specific changes.
Yeah, it often leads to GC to AT transition.
Is that right?
Exactly.
And then NTG,
NTG is a bit more potent.
more of a blunt instrument.
Kind of, yeah. It likes to target areas where DNA is actively being copied.
Oh, so the replication fork.
Yeah, the replication for the vulnerable spot.
So these alkylating agents, they add these alkyl groups,
They do.
and that messes with the base pairing.
It can, yeah. It can prevent proper base pairing or even lead to a base being lost entirely.
Oh, wow. So it can even knock a base out entirely.
It can. It's pretty powerful stuff.
Okay, and then what about those intercalating agents?
Ah, those are interesting. Think of acridine dyes,
Acridine dyes, okay.
like proflavin or ethidium bromide. Now imagine them like tiny crowbars, wedging themselves between the DNA bases.
Oh, so they squeeze in there.
They do, and that distorts the shape of the helix.
Like putting a kink in a hose.
Yeah, exactly. And that often leads to frame shift mutation.
Frame shift mutation, so that's where you either add or delete.
Exactly, and it scrambles the reading frame of the gene.
Right, it completely throws off the message.
Like a sentence that's missing a word or two.
Okay, so that's chemical mutagenesis.
That's the gist of it, yeah.
But we've also got physical methods.
Right, and this is where radiation takes center stage.
So we're talking like
Well, there's ultraviolet radiation, or UV.
UV radiation. I know that can damage DNA.
It can. It gets absorbed by the DNA, particularly those pyrimidine bases.
The pyrimidians, right?
Yeah, and the main problem is it causes these dimers to form.
Dimers, so that's like two bases stuck together.
Exactly, like two bases holding hands where they shouldn't be.
And that causes problems during replication.
It does. It can lead to base changes or even frame shifts.
Oh, right, because the machinery can't read those dimers properly.
It can't. It throws things off.
But UV radiation can also be useful, right?
Yeah, it's often used for sterilizing surfaces.
Right, because it kills bacteria.
Precisely.
Okay, and then there's the ionizing radiation.
Right, that's the big hitter.
That's the heavy artillery.
It is. It's high-energy stuff like X-rays and gamma rays.
And it can cause some serious damage.
Oh, yeah. Direct breaks in the DNA strands, damage to the bases themselves,
Wow.
and even large-scale rearrangements of chromosomes.
Oh, so it can really scramble things up?
It can. And it can also generate these reactive oxygen species.
Reactive oxygen species, or ROS?
Right, ROS. And those can cause oxidative damage to the DNA.
Oh, so it's not just the radiation itself, it's also the byproducts.
Exactly. It's a double whammy.
So compared to UV, ionizing radiation can cause a wider variety of mutations.
It can. It's less specific.
Okay, so some examples of ionizing radiation would be like...
Well, you've got X-rays, of course.
Right, X-rays.
And gamma rays from sources like cobalt-60 or cesium-137.
Oh, well. And you remember reading about fast neutrons.
Fast neutrons, yeah.
Those are used a lot in plants, I think.
They are. They have some specific applications in plant science.
So that's the physical side of things.
Yeah, there's the rundown on
But
radiation.
then we've also got this biological mutagenesis.
Right, and this is where things get really interesting.
Because now we're talking about using biological agents.
Yeah, things like transposons.
Transposons, I've heard of those. Those are the jumping genes, right?
They are. They can move around in the genome. Or engineered viruses.
Viruses. So this is like, using nature's own tools to tinker with DNA.
It is. It's harnessing these natural processes.
So these transposons, they can just randomly insert themselves into the genome.
They can, yeah. And when they land in a gene, they can disrupt its function.
Oh, so they can basically turn genes on or off.
They can, or they can create these new gene fusions.
Oh, so they can mix and match genes.
They can, or they can even change how a gene is expressed.
Wow, so it's a pretty powerful tool.
It is. It's great for discovering what different genes do.
Because you can just see what happens when you disrupt them.
Exactly. It's a bit like, throwing a wrench into the machinery and seeing what breaks.
And then what about the engineered viruses?
Well, those could be designed to carry specific DNA sequences.
Oh, so you can basically program them.
You can, and then they deliver that DNA into the host cell.
And it gets incorporated into the host's genome.
Exactly.
So what kind of viruses are we talking about here?
Well, there are retroviral vectors, for example.
Retroviral vectors, those are often used in eukaryotic cells, right?
They are.
And then there's AAV.
AAV, adeno-associated virus, that's often used for more targeted work,
Yeah.
particularly in mammalian systems.
Okay, so just to recap for everyone listening,
Yeah, let's bring it all together.
we've got these three main approaches to mutagenesis.
Three main categories.
We've got the chemical methods, where you're using compounds that directly interact with the DNA.
Right.
You've got physical methods, which is all about using radiation to induce changes.
X-rays, gamma rays, UV light,
And then you've got these biological methods.
harnessing the power of transposons or viruses,
And all of these are aimed at one thing:
introducing heritable changes in the DNA.
making those permanent tweaks to the genetic code.
It really makes you appreciate the ingenuity of all these different approaches.
Yeah, it's amazing how scientists have figured out all these ways to manipulate life at such a fundamental level.
It is, and the potential applications are huge.
Yeah, in medicine, agriculture, industry.
It really does make you think about the possibilities.
It does. It's exciting to think about what the future holds. So for everyone listening, if this has sparked your curiosity, go out there, explore, and see what kind of applications really resonate with you.
It's a fascinating field.
It is. Thanks for joining us on this deep dive.
Absolutely.
We'll see you next time.